Jai Hind everyone. I am Pankaj Kumar and hope so you people are doing well. And today's lecture is about the molecular basis of inheritance PYQs, and this is very important question. Why? From this topic in every year, like the minimum question will be six, and maximum can be eight. Okay, so in average 6 to 8 questions they will ask, but definitely six question is going to be come from this chapter, and this is the interconnected topic for the um, biotechnology chapter, principle and process. And if you will understand this chapter, it will help you in the understanding of the biomolecules nucleic acid chapter also. So that is the importance of the chapter. So this is going to be very helpful for the 2024 exam, and in this video we will solve the questions PYQs, and we'll expect what are the upcoming questions, like probable questions to be asked in the 2024 NEET examination. And nonetheless, it will going to help you in your board exams also. So stay tuned with me and let's see the questions. I will request you to first solve these questions by your own, and these questions can be available in the easily available in the uBiology app. You can go and check there and solve there. So these are the questions. I am scrolling. You can pause your video and solve these questions, and let's after that only it's good to be see the answer. Oh my god, these many questions are there. So I took questions from the 2029, and you will understand the trend of the NEET examination where it is shifting. If you will calculate the marks, it will give you around 32 marks, or around 32 marks, and this 32 marks out of 720, so it will be around 4% of your complete weightage of the NEET. And if you see in the biology, it will have the 8 percentage. Okay, and only in biology section it will have 8 percentage of the weightage. So first question is from the purine from 2029. The question is that purine found both in DNA and RNA are cytosine and thymine, adenine and thymine, and adenine and guanine. Okay, so what is the difference between the RNA and DNA? RNA and DNA. You must remember this is deoxy, means there is a lack of the 2 prime OH, but here there will be 2 prime OH will be there in this RNA, and there will be absent. And second thing is that DNA have a thymine, and instead of thymine, RNA have a uracil. And adenine and purine and pyrimidine is what? Purine is something which is called AG, adenine and guanine, and pyrimidine is something which is called cut, cytosine, uracil, and thymine. Okay, so what they are asking? Purine found both DNA and RNA. What are the purine? A and G are the purine. So it will be present in both A, DNA and RNA. Only thymine is absent in the RNA. Instead of thymine, it have a uracil. What will be the sequence of mRNA produced by the following stretch of DNA? So sequence of mRNA they are asking, produced by the following stretch of DNA. So this is a stretch. This is called as a template and coding stretch. So remember it. Whenever they will called as a template on which on which template on which the new mRNA is going to be formed, and coding means in which RNA will not form. Okay, coding strand. Now it have another name also. Template is also called as a non-coding, non-coding, antisense, sense, and negative strand. Now coding will be called as a non-template, non-template, and coding strand, sense strand, and positive strand. Okay, so this is the important different names, but you preferably you remember these two names, template and non-template. Okay, so template is the structure on which frame structure on which the new mRNA is going to be formed. So this is a 3 prime to 5 prime. So new mRNA will form from the 5 prime to 3 prime. Got it? So it will form as a 5 prime to 3 prime. The one more similarity, if you'll see, observe properly, this is also 5 prime to 3 prime. Yes or no? Coding strand also 5 prime to 3 prime, and mRNA is also for this is mRNA, and this is also mRNA is also 5 prime to 3 prime. What do you think? This will be both similar or different? You can comment in your chat box. This will be both similar or different. Definitely it is going to be similar, but the minor change will be there. This is a coding strand is a DNA strand, and this is a RNA. So RNA will have uracil and DNA will have thymine. So mRNA will be exact copy of the coding strand, except the instead of the thymine it will have a uracil. So you just make a copy directly from this. What is this? T. The prime will be same. 5 prime is here. 5 prime is here. So you find what are the 5 primes? 5 prime to 3 prime. This. Okay. There is a T. It should be U here. So this is not your answer. This can be your answer, and this can be your answer. But it cannot be your answer. Okay, because it started from the 3 prime, and it started from the 5 prime. Okay, but here it should be adenine. Here it should be adenine. Okay, so T will bind with the T will be complementary to the adenine. Got it? So here it is A. Here it is A. So what could be your probable answer? It will start with the 5 prime. So D could be the probable answer. But we will match complete sequence. Okay. So T will complete. Sorry, you just replace this all T. What are the T's are there? You this just replace all T with the U in the 5 prime strand. This is the T. This is the T. This is the T. This is the T. And okay. So now you replace it with the uracil. Okay, you replace it with the uracil, and then you match UACGU. Okay, then ACGU A. Okay, 1, 2, 3, 4. Fine, it is going correct or not? So it is going to be correct only. U A C G. There is a one U. After that, there will be after G there will be another U. A C G. Then one A C G. Here this one. U 1, 2, 3, 4, and then ACG again U. So we are done with it. This is your answer. So actually, after knowing the answer also you may get confusion because of this. You are solving in the question only. Better what you do? You write separately here in separately here somewhere in the page, and then replace the coding strand T with the U. That will be your mRNA. Okay, important thing. What I replaced? I replaced the T from the coding strand. You cannot replace the T from the template strand. Got it? This is the important thing. Okay. Second thing, you have to match the prime. It should be 5 prime to 3 prime. Okay, it should be 5 prime to 3 prime. So answer will be your, it should be 5 prime to 3 prime. Okay, this will be your answer. Hope so you understand this. Next, match the following mRNA RNA polymerase with their transcribed product. So RNA polymerase 1 is for what? RNA polymerase 1 for the rRNA. Okay, rRNA is the abundant RNA which is around 80%. It is forming the ribosome. Next is what? Next is RNA polymerase 2. It is going to form hnRNA, and further after processing, it will give mRNA. Got it? Now tRNA will be produced by the polymerase 3. So it is also producing the tRNA and 5 prime rRNA also, and snRNA also. So okay, we'll see the matches. One is matching with what? One is matching with the 2. So where it is, one is matched with the 2 here, and this could be the one of your answer, correct answer. 2 RNA polymerase 2 with hnRNA and 3 RNA iRNA. Okay, fine. Under which of the following condition there will be no change in the reading frame of the following mRNA? So when will be the reading frame will be not changed? What do you mean by reading frame? What do you mean by this reading frame? So reading frame means what? Whenever we'll read the genetic code, it will be read in the triplet form. Okay, it will read in the triplet form. Okay, so it will be like if you want to read this, you will read like AAC, then AGC, and then GGU. Like that, you will read this complete reading frame in triplet. So the frame will be changed if you will insert any letter. Suppose if I am inserting one letter, one adenine here. So what will be the frame now? The frame will be changed from here to A C. This is the first is same, but the next here it will change. It will become now A G, and then this will goes with this, and then it will become C G G, and then U will be remains. Got it? So whenever we are adding or removing, or if you will delete any codon, then the reading frame will be changed. But what if I will completely remove this one? So they are asking if you'll remove this GGU at 7, 8, 9 position consistently. Okay, so the reading frame will be same only. You're not going to get the new reading frame. So what will be reading frame? So see, now it will be ACC, AAC, I'm erasing it now, AGC, and now this is empty. So these both are correct. Afterward, it will continue like that. So reading frame will not change if you delete the triplet. Okay, triplet codon. So answer will be the A. Which of the other things I mentioned here, insertion or deletion, one or two will be the affect the reading frame. Which of the following feature of the genetic code does allow bacteria to produce human insulin by recombinant DNA technology? So this question is from the recombinant DNA technology, but uh, not from recombinant DNA technology, but it's both connected. In beginning only I said chapter. So genetic code is specific. Genetic code is not ambiguous. genetic code is redundant and genetic code is nearly universal. So it is the correct answer. Genetic codes are universal. Either it is in the human or either it is in the bacteria. AUG will always code for the methionine. Okay. Or UUU, wherever it will be, it will code for the phenylalanine. Like that, we have a 64 genetic codes, and that will be universal, nearly universal to the every animal. Non unambiguous means what? One genetic code can code for the one amino acid, but cannot code for the other amino acid. Redundant means what? It is the also called as a degenerate codon. Means what? One amino acid can be coded by more than different amino acid. Okay, one amino acid can be coded by more than different genetic codes. But unambiguous means what? Again, I'm repeating. You can write in your notebook. Unambiguous means one codon will code for the one amino acid. Degenerate, one amino acid can be coded by multiple codon. Okay, better if I will give you an. So here are some examples you can see here. ACU or ACC, ACA, ACC, ACA. This all will code for the threonine. But this all are called as a degenerate codon. This all will called as a degenerate. But only ACU or any ACC or ACA is coding for the one amino acid. So ACU is called as a unambiguous. Unambiguous. Why it is called as a unambiguous? Because it is not going to code another amino acid other than the threonine. So it will code always threonine. It will code always threonine. So it is unambiguous. Okay. And this why it is called as a degenerate, because all having a same love with the threonine. All will have the going to code for the threonine. Okay. This is done. Next question is what? From the lac operon. So I gene is coding for the repressor, you remember, and Z gene is coding for the beta galactosidase, A gene transacetyl is Y permease. So if you want to read the lac operon, the link is given here in the above line. You can go and watch that lac operon. And all the chapters PDFs are available, notes available in the U Biology. You can go and download there. You can read there. If you need any kind of topics, lecture or videos, you can text me there in the comment box, and definitely I will provide. So according to this, I gene is a repressor, A with 3, Z gene with the 1. Okay, so this will be eliminated. These questions are very easy to answer correctly. Okay, so this question should not be going to wrong, because easily you can eliminate the option. Got it? So I eliminated these two. Now see, A gene is transacetylase. Okay, if you don't know about A gene, then at least you know about Y gene. Then you will find Y gene is for the permease. So this will be your answer. Express sequence tag is referred to the what? It will refer to the gene expressed as a RNA novel DNA sequence and the polypeptide expression DNA polymorphism. What these questions are telling, like sometimes you will not understand what is the polymorphism or two options. So don't worry about that answer. You just focus on the option in which you are confident. Okay, so express sequence tag means this DNA region which is going to express as a RNA. Got it? That is called as DNA, genes expressed as a RNA. Polypeptide expression, polypeptide means what? Protein. Okay, so it is not going to only produce the protein. DNA polymorphism means different DNA we have, like from you and me, from your mother and father. So everybody have different polymorphism. and it will help in the gene mapping. Okay. Why this polymorphism is there? Because of repetitive DNA sequence. So this is the question from the 2029, and we have to increase our speed, I think. Definitely. Now, so now this is the question from the 2020. So 2020 questions are there. The first phase of the translation is binding of mRNA. It is called mRNA to the ribosome. No. Recognition of DNA molecule. No. Recognition of anticodon. Amino acylation of tRNA. This is the correct answer. What do you mean by amino acylation of the tRNA? The activation of tRNA. Okay. Activation of tRNA is called as the amino acylation. So you can see here, these are the lag operon system, which I explained in the videos. Translation. So a small, where it is, let this stop here. Huh? Now, so tRNA was discovered by the Holly, and it have a two shape, D form clover leaf like and 3D L shaped. So whenever the amino acid will bind to the 3 prime by the help of ATP, remember it, okay, by the help of ATP, then it is called as a activation of tRNA or amino acylation of tRNA. Let's come to the question again. So the answer is amino acylation tRNA. If the distance between the two consecutive 0.34 nanometer, then total number of. So if you want to calculate this, so the length of DNA, you can calculate like that. Length of DNA helix will be equal to the two consecutive base pair distance between two base pair. Okay, two consecutive base pair. Okay, multiplied with number of base pair will give you the complete length of DNA. If you multiply, and you have to remember the unit should be same. Unit should be same, and you will find that it is 2.2 meter. If you multiply and you will calculate. Enzyme. Name the enzyme that facilitated the opening of a DNA during the transcription. So either it is a DNA transcription or replication. The DNA helicase is the important enzyme which is going to be open the DNA. So here they are asking enzyme facilitated the DNA helix to during the transcription. So DNA helix will be open in the DNA replication. Ligase is for the sticking, for forming the phosphodiester bond. It will stick the two nucleotides, and then DNA polymerase is for the polynucleotide elongation, and answer should be RNA polymerase. So it will behave. It is a single independent RNA polymerase which is going to form the opening the DNA and elongation and then termination. So it is kind of independent type of RNA. It doesn't require several units. So DNA polymerase is the answer for this question, which is going to be open the DNA. Which of the following statement is correct? Adenine pair with the thymine with through two H bond. So whenever you'll find the first answer, you don't have to go direct. You don't have to tick the answer in your question bank. First, you highlight what they are asking, correct or incorrect. So they are asking correct. So even you found this answer, please read all the options. Adenine pair with the thymine through the one bond. Adenine pair with the thymine through the three bond. Wrong. Adenine does not pair with the thymine. This is wrong. This is wrong. This is wrong. So adenine bind with the thymine with the two bond. So in this year only 2020, they asked 4 questions. After that, they asked like 6, 7, 8, and complete the flow. You just solve this question and then tell. DNA replication. This is the replication, then transcription, and then translation, and this is protein. So what will be your answer? Replication, transcription, transduction. No, this is wrong. Replication, transcription, translation, protein. Okay, this is the correct answer. What do you mean by transduction? If you don't know, please comment me there. So whenever the bacteria will infect the infected by the viruses, that's called a transduction. Identify the correct statement. Now you again highlight what they are asking. They are asking correct. So now let's see. Capping methyl guanosine triphosphate is added to the 3' end. Is it so? No. RNA polymerase, it bind with the Rho factor to terminate the process of transcription in the bacteria. It seems good, but we'll read all. The coding strand of the transcription unit is copied to an mRNA. Just now we seen it's not the correct answer. Template strand will be copied to the mRNA. Okay, I given you there as a hint to easily solve the questions. Okay, but template strand will form the mRNA. Split gene. What are the split gene? Split genes arrangement is characteristic of prokaryotes. So like exon and intron. Intron will called as a split genes, and then if gene will have the intron, that's called as a split gene. But important thing, that is a property of the eukaryote. Prokaryote doesn't have split genes. Introns they don't have. Usually we can say. So correct answer is what? This second. Now, what is the role of the RNA polymerase in the process of transcription in eukaryote? RNA polymerase 3. So, RNA polymerase 3 is going to form the tRNA. Where is the tRNA? rRNA, tRNA, precursor mRNA, snRNA only? No. So, it is going to form, transcribe the RNA. This is an important thing. They can ask what 5S RNA is produced by what? rRNA, polymerase 1, 2, or 3. So, this is the only rRNA which is synthesized by transcribed by tRNA. Now, important thing is that tRNA and rRNA is called as a long non-coding RNA. Okay, it's not going to code anything, but coding RNA is mRNA. Now, DNA fingerprinting involves the identifying the difference in some specific region of a DNA sequence called. Okay, now what is satellite DNA, repetitive DNA, single nucleotide, and polymorphic? Satellite DNA is the DNA which have the repetitive DNA. Okay, so you can see primary and secondary satellite. Okay, you must have seen primary and secondary satellite. Okay, so primary satellite centromer is one centromere is the satellite. Okay, so what it have? It have a repetitive DNA which is very common. Now this repetitive DNA is going to give you the V and TR. Okay, now this satellite have a micro and mini satellite. So micro and mini satellite means it is going to give you the V and TR, variable number of tandem repeat. You must have read, and this is used for the DNA fingerprinting. So now these questions and options are like more similar, but what is the most important concept? Is the satellite is the important, or centromer is important, or repetitive DNA? So answer is the repetitive DNA. If I'll ask the building formed by what? Building formed by the rooms or floor or bricks. What will be your answer? Answer will be brick. So repetitive DNA is the more fundamental thing. So answer will be the repetitive DNA. Polymorphic DNA. Because of repetitive DNA, we have a different type of DNAs in you, me, and all. Like 99% of the DNA of all human beings are same. Just 1% is different. Why it is? It is because of repetitive DNA. And that makes polymorphic DNA, like difference in the pool of a DNA in a population. Which of the following RNA is not required for the synthesis of protein? You tell me. tRNA required, mRNA required, rRNA required, siRNA. siRNA is not required. So this is the correct answer. Do you know what is the siRNA? Why it is? So till now you know the DNA will form the RNA and RNA will form the protein. But siRNA is the long non-coding like small interference. It's a non-coding RNA which will form as a RNA induced silencing. RNA induced silencing it is called. RNA induced silencing complex. So it will silence the mRNA. So it will regulate the RNA like protein synthesis. So it is called as a risk. What it's called? Short form is risk. Now, which is the only enzyme that has a capability to catalyze initiation, elongation, termination? So it is a DNA dependent RNA polymerase. Just now we seen in the above questions. So these are the questions which is repetitive and asked in the different form. They ask in the opening of the DNA, which is important. So now they ask which can do everything by itself. Which of the following statement about hormone is wrong? Okay. Now tell me. They are asking wrong, then you highlight it. Like what they are asking, correct or incorrect, wrong, correct, highlight. Histone are organized to form unit of 10 molecule. The pH of histone is slightly acidic. Histone is rich in the lysine and arginine, and histone carry a positive charge in the side chain. Okay, so what will be your answer? So histone is lysine and arginine are the basic amino acid. These are the basic amino acid, not acidic. What are the acidic amino acid? Glutamic acid or aspartic acid. So this is the wrong answer. This is the wrong statement. So answer will be this one. What are the histone proteins? Like H1A, okay, H2, H3, H4, H1, then H2A, H3, H4, and here you can write one more. 1, 2, 3. Okay. Then H2B also you can write. And H2B, H3, H4. So this will form a nucleosome, and this will form a core, and this will form as a linker DNA. It will be connected by the link. We'll see this nucleosome. So H1 is like this, and then these are the 4 DNA: H2A, H2B, and then H3 and H4. So whenever the DNA will come, DNA will be wrapped from here, and then it will go for this. So this will call as a nucleosome. It have what? How many DNA is wrapped around this? How many nitrogenous base? It will be around 176, and from here to here it will be around 146, I think. From here to here it will be around 200 base pair. Okay. So this is, you remember, this will form as a it will called as a octamer. It should be 8 only, na? Why we form only 4? So total there will be copy of the DNA. Sorry, histones. To this will be 2, each having the copy. So total 4 have 4 into 2, 8 complete octamers will be there. Now read the following statement, choose the correct statement. Again you have to underline what they are asking, correct statement they are asking. Okay, so let me go little fast, and if you need any explanation further you can ask me in the comment box. So euchromatin is a loosely packed chromatin Correct only. It is loosely and it is a expressing one. It will express. Sometime I'm not writing. Please write it on your notebook. Heterochromatin is, it will be dense. It will be dense and it will be transcriptionally active. It is not going to form any RNA, so it is transcriptionally inactive. Histone octamer is wrapped by a negatively charged DNA. Negatively charged DNA in the nucleosome. Histone octamer is wrapped. Histone octamer is wrapped by negatively charged DNA in nucleosome. Histone is rich in lysine and arginine, and atypical nucleosome contain 400 base pair of a DNA and DNA helix. So among this, we have to find the options given below. So A is correct. Histone is the octamer. This is correct. Wrapped by negatively charged. DNA is a negatively charged, and this arginine and lysine are the positively charged. Okay, these are the positively charged amino acids. Histone rich in the lysine and arginine. This is also correct, and this is wrong. So A, C, D is the correct answer. Okay. Now you can see every options, but I have to go a little fast, please. DNA polymorphism form basis of what? DNA polymorphism is what? It is because of repetitive DNA, and repetitive DNA help in the DNA fingerprinting. So after seeing the DNA fingerprinting, don't read all the options. Okay, just as I said, both genetic mapping and DNA fingerprinting. Translation, no, genetic mapping. Okay, so DNA, it will help in the both genetic mapping and the DNA fingerprinting. What is the meaning of genetic mapping? So you know there is a X chromosome, and X chromosome is for the female. You know now how we found this, and Y chromosome is for the male. So by because of genetic mapping. Okay, you know because of 21st chromosome of the uh, gene, 21st chromosome triploid uh, will form a trisomy, will form the Down syndrome. Okay, 21st or 18 will give you the uh, will give you the Tau syndrome, Edward syndrome. So this is because of the their chromosome number, and there will be sighting. This is a complete big DNA. This, suppose this is a gene for the hair. Okay, this is the gene for the height. So how we will find this all things? Because of the mapping. Okay, so we'll found the repetitive DNA site, and then we'll map the DNA and their chromosome and their function also. So it will help in the DNA mapping, and you know how it will be used for the DNA fingerprinting. So you can scroll the videos in my playlist. You can find it there, the complete explanation. The process of translation of mRNA to protein begin as soon as the large subunit of ribosome encounter mRNA. Both subunits join together to bind with the mRNA. Uh, the process of translation of mRNA to protein begin as soon as. So when the translation of mRNA will begin. First step, you know, the amino acylation of a tRNA. The tRNA activated and the large subunit ribosome encounter. This is tRNA activated. This is correct, but first it will be not going to be bind with the large subunit. It will bind with the small subunit. So small subunit ribosome encounter mRNA in the following palindromic base. Where it will bound? This is called as a this will called as what? Called as a Sandelgerno sequence. Okay, SHINE Sandelgerno Gerno sequence. DNA, which one of the can be cut easily by particular restriction enzyme. So this is one of the biotechnology, but you can remember it. GATTC is the Ecori site. What is the meaning of palindromic sequence? So palindromic sequence which have a repetitively, like from uh, 5 prime to 3 prime, if you'll read, or you will read 3 prime to 5 prime, it should be same. So they given the Malayalam is a good example. Okay, or I will write uh, like this. So if you read from the any side, it will called as a Malayalam like that. If you read from the 5 prime to 3 prime, GAATTC, and if you read from the 3 prime to uh, 5 prime, again it will get GAA GAATTC, again GTATTC. You can check CTCAGT. No, it is not. GTATTC. No, this is also not matching. GATA. So GATTC is your palindromic sequence. Got it? So transposon can be used during which one of the following? Transposon. What is this transposon? It will called as a jumping gene. Okay. And what is the use of this jumping gene? So I will tell you, suppose this is a gene for the height or for the RBC. Suppose this is the gene for the RBC. Now this is another jumping gene which is somewhere here. Got it? Now it will be jump from here and it will come in between this. So if the jumping gene will come between this, then our RBC gene will be change. Yes or no? Because it is interrupted by the jumping gene. This is interrupted by the jumping gene. Now whenever the polymerase will come, it is not going to form a RBC required gene. Okay, it is not going to be formed. But here it is easily can form the RBC. Got it? So jumping gene will silence the gene. It will help in the gene silencing and like that. Autoradiography, gene sequencing, no. Polymerase chain reaction, you know. Autoradiography, autoradiography you read in the biotechnology chapter now when you will going to visualize the DNA electrophoresis. Okay, that will use for the imaging. Gene sequencing, the sequence of the gene. Polymerase chain reaction, you know. In if a genetist used blind approach to sequence the whole genome of the organism, like uh, like you people are the genetists suppose, and you want to sequence a genome followed by assignment, and you have assignment of the function of a different segment and the methodology adopted by him, and you have to find the complete sequence of a gene. Suppose this is a chromosome, you have to find the sequence of it, and you have to find what is the function of this. You have to find the function of this gene also. How you will do? What will be the important thing? Think what you have to do. You'll do gene mapping, express sequence tag, bioinformatic, sequence annotation. Sequence annotation means what? Uh, Assigning the sequence and telling the what is the sequence and assigning the function also. So I told by gene mapping only you will map, then it is fine. In mapping you will find the position like what is it? A G G A C T G A C like this. Okay, but by that you will not find. You have to find the function also. So that is called as a sequence annotation. Okay, that is called sequence annotation. Okay, length of the DNA molecule meter, approximate number of the base pair. That you have to tell. You can calculate and you will find answer is 3.3 into 10 power 9 base pair. Before that also we see. This is the important question. Like you have to find the 10 E. coli cells with 15N double-stranded DNA and incubated in the medium of containing 14N nucleotide. After 60 minutes, how many E. coli will have the DNA totally free from 15N? So you can try by yourself. I'm going to solve here. You pause the video and you just find. So first, the few important thing you have to know before solving this question. First thing is what? The 60 minute. How many times the cell will divide in 60 minutes? So the E. coli will divide into 20 minutes. In 20 minutes, one division. Okay. So in 60 minutes, how many division will be there? So 60 by 20. So the cell will divide 3 times. Okay. So 3 divisions. Cell will divide 3 division. Got it? Okay. Hope so you understand this. Now this is the first division, second division, and then third division. So in one division, how many cell you will get? From one cell you will get 2. From second division, from 2 you will get 4, and from 4 you will get 8. Let's see. I am going to let's see how many N14 cells are there. This is the Martha and Chase experiment. Martha Chase and Hershey and Chase experiment. Sorry, Hershey Chase experiment, and then we are continuing this. The red color is for the N14 strand, and blue color for the this N15. So they provided N14 medium in this. Afterwards, there will be always N14. There is a no N15 medium nucleotides. Okay. So whenever it will divide, this two strand will be divided into this way, and one strand of N15, one strand of N14 you will get. Okay. So in this case, I am hydrogen bond I am making with the red only, so don't worry about it. Just simply I am making. So this is the two strand. One is N15 and one red is the red is what? You tell me. Red is what? N14. Good. And blue is N15 strand. Now it will make a copy, and now hence we don't have N15, so and no N15 new N15 will come. Only this two will be N15, and then all the strands will be N14. Okay. This is the DNA I'm going to making here. Now this all strands are N15. Got it? So how many cells are there in this? In second generation, how many cells are there with the N15? The first, second, 3, 4. So first and 4 is the only N15. Okay, first and 4 is the N15. Like that, if you'll make here, if you'll go for the second generation, third generation, then what you will find? Only 2 will be the N15, remaining will be N14. Okay, you don't have to do these many things. I explained you for this explanation. So let's see how many N15 you can find. How many cells have the N15? Only 2. This one is the N15 and this one is the N15. Remaining are all N15. So 2 are the N15. Okay, and 6 are the N14. Is it for the 10 cell or for 1 cell? So we took here only 1 E. coli. Now in 1 E. coli, this is 2 is N14, N15, and 6 is N14. Then 10 in 10, how many it will be? So you multiply with this. What they are asking? They are asking how many E. coli cells have DNA totally free. Important. This is free from N15. You don't tell in which N15 is present. Totally free. So you multiply with 10 because this is the result for one cell. Multiply with free means what? Only N14. So multiply with this. You will get 16 E. coli. And how many total cell you will get? Multiply with this, and then you will finally add 16 and 20. Total 80 cells will be formed, and 60 will be the equalize cell with N14 only, free of the N15. Got it? So they ask free, not with. Okay. Now 2023. So I think recording is going very long. I will suggest to watch this till end. Okay. Unequivocally, proof of the DNA genetic material was first proposed by, like, without any doubt and without any uh, controversy. This is the Alfred Hershey and Chase experiment. Okay, sorry. This was the Michelson and Stahl experiment. Okay, this was continued. This N15 and N14 was by the Michelson and Stahl and Stahl experiment. And this Hershey and Chase they took as a bacteriophage and the bacteria. Then they S35 they took, okay, and P32. Express sequence tag, again they are asking the same question. Refer to all gene that expressed as RNA, all gene that expressed as a protein, all gene expressed as a unexpressed certain important expressed gene. Answer will be expressed as a RNA. What is the role of the RNA polymerase 3? This, remember it? 3 is for the tRNA. Again, this is the second time they are asking, or third time, I think. Same question. This is the RNA polymerase function are very important. In prokaryotic, positively charged DNA is held with some negatively charged protein in the region called nucleoid. So the same concept is there. Nucleosome concept, histone concept is there. So this is also important concept. Histone and nucleosome. They are asking in the third times at least. So nucleosome having a lysine and arginine. These are what? Positively charged, not negatively charged. So this is the wrong statement. In eukaryote, negatively charged DNA is wrapped around the positively charged histone octamer. Charged DNA. So DNA is what? Negatively charged. Okay, this is not positively charged. So answer is what? Statement 1 is incorrect, 2 is correct. Gene A is for what? Gene, this is also important, like lack of proton. Gene A is what? Uh, Acetylase, trans acetylase. So A with 2. A with 2. What are the two options? Now eliminate these two and go with first and 2. Now B, Y is what? Permease. So 3. If you don't know Y, then check for the C. Gene I is for the repressor gene. Now this is both same, then you check for the D. Z is for what? Beta galactosidase. Okay, like why I'm saying to check for the another. Sometime if you don't know one, then you will find and get an idea from the other options. So this match the following should not be wrong in for any physics, chemistry, and biology. Which of the following is a sequence on corresponding to the coding strand? If the sequence of mRNA should is as follows. So now they given the mRNA sequence. What they given? This is the our mRNA. Okay, and what they are asking? This is our mRNA, and they are asking what? Coding strand. What is this coding strand? Is it going to code the mRNA? No, not at all. Template strand is going to code the RNA. Non-coding strand is what? Non-template. Okay. So just you remove, you just replace this U with T, then you'll get the coding DNA, because coding is in the DNA. This is in DNA, and DNA doesn't have uracil, so you replace this T. So priming will be all same, 5 prime to 3 prime only. Just see 5 prime to 3 prime. Then A T. Okay, this will be then A T. Here also T. A, T. Okay, all same. You can count also. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. Okay, this is the correct answer. Now, recently last year question. So, transcription unit in DNA is defined primarily by the three region of a DNA. What it is? So, upstream and downstream. Considering upstream and downstream. So again, this is from the lack of brown, but uh, question is little bit changed. What are there? Repressor gene. It can be. It cannot be. It's not a part of the transcription unit. Okay, what is there in that? So whenever I will talk about the transcription unit, there should be one promoter which will promote the structure. Then there should be some gene which is going to be produce the RNA, and then this is there should be one terminator. Okay, this is the ideal thing. Now whatever it is there, operator it can be here, okay, but it cannot be, and repressor gene will be somewhere away from this. This is upstream and this is downstream. So this is our unit. Now promoter structural gene is important. Transposon is not there. Operator gene no. Inducer no. Repressor no. These are the matter. It's not a gene part. Okay. So promoter, structural gene, and terminator are the important thing. Now again lack of proton only they ask here. A lactose present in the growth medium of a bacteria are transported in the cell. So if the lactose is there, this is a bacteria and it have a so much of lactose outside this. So how it will take this lactose outside from this? So it required one channel, and that channel is produced. That channel is like a mouth, and this channel is produced by the primase. This is by the primase channel. Okay. So this all will be taken by the primase. So this will be the correct answer. Match the following. Frederick Griffith. What they did? Transformation. Transformed the rough bacteria into smooth bacteria. Streptococcus pneumonia. Okay. A is 3. A is 3. A is 3. So now 3 options are there. Better what you do, what are the difference in that? B have all different options. Okay, B have all different options. So you directly check B. Francis, Jacob, and Monad. Lack of 4. Where is the B with 4? Here only B. So you can save the time, but again it depends upon your practice. So I will ask you to read and follow the ones all the options. Once you will find this, then match A. A is transformation, correct. C is 1. Hargobind Khurana is genetic code, correct. D. Messelson and Stahl. D. To semi-conservative mode of DNA, correct. DNA present in the chloroplast is, so it is, you can find it as a question from the cell chapter, but it is also important like what are the type of DNA is there. So DNA can be linear and double stranded. So eukaryotic DNA are the linear and double stranded. So chloroplast is a eukaryotic part. It is present in the eukaryote cell, but it have a different type of DNA. It is a circular and double stranded. So answer is what? This. Which of the following? I'm saying you have to read all the options. Which of the following statement is correct regarding the process of replication? So DNA dependent RNA polymerase catalyzes polymerization in one direction, that is 3' to 5'. The DNA dependent uh, 3' to 5', no. DNA dependent RNA polymerase catalyzes the polymerization of in one direction. This is 5' to 3' direction. This is correct. DNA, they are asking correct statement. DNA, the DNA dependent DNA polymerase catalyzes polymerization in 5' to 3' direction as well as no, it will always polymerize from the DNA dependent DNA polymerase catalyze polymerization in 5 prime to 3 prime direction. Now DNA dependent DNA polymerase catalyze polymerization in 5 prime to 3 prime direction. So what could be the answer for this question? This DNA dependent DNA polymerase and DNA dependent RNA polymerase catalyze polymerization in one direction, that is 5 prime to 3 prime direction. DNA dependent DNA polymerase catalyzes polymerization in 5 prime to 3 prime direction. Oh, little tough. Why it is? Because these two both options are correct. Now at this time you have to like think with you what you have to go with it. You can challenge it, these questions. The DNA dependent RNA polymerase catalyze the polymerization in one direction, that is 5 prime to 3 prime direction. That is correct. DNA dependent DNA polymerase catalyzes polymerization in 5 prime to 3 prime direction. Polymerization in one direction. So both options are clear. So we have to, if we have to choose, then we have to choose more appropriate. Hmm. Let's see. Let's come with your opinions in the comment. Then we'll discuss there over. So, which one is the correct product of the DNA dependent RNA polymerase to given template? So, it should be, I think it should be more appropriate for the fourth option from my side. But let's see what is your option you're going to give. Because this is not only just one direction, it's in single direction, but more appropriate DNA dependent DNA polymerase catalyze the polymerization in 5 prime to 3 prime direction. Which of one is the correct product of a DNA dependent RNA polymerase to given template? 3 prime to 5 prime. This is the template strand. Now this is the good question. So this is the template strand. You have to find the correct product of a DNA dependent RNA polymerase. So you have to find the RNA mRNA sequence. Okay. Now this is a template. Then you have to find the mRNA. So template will give you 5 prime to 3 prime mRNA. Okay. This will be same as a coding only strand, but instead of T you have to write A. So here it is a T first. It is a A. Then A will bind with the T, but T will replace with the U. And if you'll find a difficulty now, I will tell you simple thing. If it is a DNA, just write the DNA sequence. You match T will bind with the A will bind with the T, C will bind with G, A will bind with T will bind with A, then C, G, T, 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 A, then G. Now this is your DNA strand. This is again I'm saying this is your DNA strand, DNA coding strand. Okay, now you replace T with the all the T with the U. Okay, then it will be your answer. Okay, so your answer will be what? 5 prime to 3 prime. A U G U A C C G U U U A U A G G U A G U 3 prime. Okay, now you find it answer. So whatever matches with it, you can find it. G G A U. G double C. Okay, this is your answer. First option is your answer. You have to match by yourself. Now, RNA polymerase 3. Again, this is the very important thing. You must remember this for your NEET examination. RNA polymerase 3 bind going to snRNA, tRNA. Okay, correct. So A with 4. A with 4 only. There is only one option, so you can go with it, but you check everyone. Now, if you find this 4, then you check B and 3. B bind with the termination of transcription will be done by rho factor. This is also correct. C splicing of exon by snRNPs. That is also correct. And then D Tata box is there in the promoter region. Tata box. So what is snRNP? It is a small ribonucleoprotein. Small nuclear ribonucleoprotein, and it will help in the splicing. Okay, splicing it will help. There are different type of snRNP like U1, U2, U7 like this. Okay, fine. You don't have to go much deep, but you remember it will help in the splicing. What is the Tata box? So snRNA we already seen. RNP and RNA both are different. snRNP it is protein which is help in the splicing. This is the RNA. Now Tata box is what? This is a DNA, and this DNA will have a tata repetitive Tata box. Okay, and then this sigma factor you must have remembered in the transcription. This sigma factor of a RNA polymerase it will bind here, and it will recognize, and then it will recruit the like complete RNA polymerase, and then it will start the transcription. So it is a recognition site, okay, for the sigma factor. So hope so you enjoyed this video, and most important, uh, I need motivation, and because of your comment and by your subscribing and by your sharing the videos, by more you'll ask me the question, more I will answer. It will motivate me to continue this video, and if you like such type of video, please do subscribe and give the comments, so I will continue such type of video. Otherwise, uh, if I will not get a motivation from you people, then it hard to be continue with such type of video. Then I will think for the different type of content. Okay, thanks for watching. We'll meet in the next video. Jai Hind. Okay, most important thing. So what are the 2024 expected questions? So see, the trends are like this. In beginning, they ask much about the RNAs and some other DNA fingerprintings. But now trend is regarding the lac operon, and most important trend which will be not going to be changed, the RNA polymerase. Okay, RNA polymerase its type, especially its type. Okay, then they asked DNA fingerprinting. What you have to understand what is the DNA polymorphism and repetitive DNA. Okay, so express sequence tag. Okay, I should write repetitive DNA. And that's all. This is the important topics which you have to ask. They didn't ask much question from the transcription replication. They didn't ask much question from the Griffith, Hersey, Martha, Hersey and Chase experiment, and they didn't ask about the MacLeod McCarthy experiment. They didn't ask about the uh, other things what DNA human genome project much. So sometimes they will ask the calculation and Michelson and Stahl experiment. Sen Stahl experiment, and you have to read about the DNA. That's it. If you know this much, then it's enough. Translation, from translation, they are not willing to ask you much deeper question, but at least you understand what is the initiation of a translation. Initiation of a translation. Okay. But if you know everything, that's very good. But these are the most important questions, expected topics, which is going to be asked in the upcoming exam. Thanks for watching. We'll meet in the next video. Jai Hind.